Go.
Hi, I'm Tamara Johnson.
I'm Kendasi Silva.
I'm Joylise Mitchell.
And this is our replica of DNA replication. DNA replication is the process of producing two identical replicas of DNA from one original DNA molecule. Asexual reproduction is the ability of cells to make identical copies of their DNA before cell division. DNA replication happens during mitosis. It is replicated during the S phase, which is the synthesis phase of interphase. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides, which are hydrogen bonded to each other, forming a double helix, which is shown as so. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of four nitrogenous bases. The phosphodiester backbones consist of alternating sugars and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine. And thymine forms 2 hydrogen bonds with adenine. This is known as complementary base pairing. Complementary base pairing allows replication to be executed in a semi-conservative way. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated in each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. Replication occurs in a 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order for replication to proceed, a number of enzymes are needed once the replication fork has been established. Helicase separates the strands of the double helix. Single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Proteins such as beta clamps and the clamp loader help hold the DNA polymerase in place on the DNA. Primers, which are short sequences of RNA, have to be paired to the template strands by the enzyme primase. This is because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occur at the same time. One uses continuous synthesis while the other uses discontinuous synthesis. Continuous synthesis occurs on the 3' to 5' parent strand, the leading strand. New nucleotides are added to the 3' end, moving continuously towards the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is 5' to 3', called the lagging strand. It is completed in segments called Okazaki fragments. Primase is used to add primers ahead of the 5' end of the lagging strand. DNA polymerase III adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, the process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments, as well as the replaced nucleotides. Once the leading and lagging strands have completed their replication, two identical copies of the DNA molecule result.